So what we had there is exposed parts of a natural forest, a mixed forest. We catapulted that forest into the future by fumigating it artificially with CO2, thus creating an atmosphere of high CO2 as we would expect it around mid to late century. So yeah, welcome to my talk. I thought I make it a bit of a research journey, so it will spill a little bit over the tight frame that I stuck here, because when I thought about what I want to present, it started to grow tentacles and arms and wings and got a bit out of hand. So it's got a little bit more than only carbon and water relations. So I thought I'd start off with giving you an overview who I actually am and my research history, and pick a few highlights from those stations. So I started off with a master's in geography first in Germany, and then more or less in parallel a master's in biology, also in Germany, and then went to Switzerland to do my PhD there on the responses of forests to elevated atmospheric CO2. So I'm going to talk in more detail about that in a minute. From there, I went on to Western Australia, where I worked on drought effects in Banksia woodlands, highly diverse ecosystems. And then I hopped over the pond to New Zealand, where I worked for the equivalent of what is Skogforsk in Sweden, the New Zealand Forest Research Institute, for roughly 5 years, and then joined Auckland University of Technology as a senior lecturer in ecology, and started working, or the research that I'm doing today started shaping out back then. And then I came to Linneus University as a professor in, well, officially forest ecosystem services, but it's broadly basically forest ecology, and where I work on forest nutrition now as well, on top of the global change topics, and also with my new colleagues from remote sensing and plant pathology on Franz and Johanna Witzel on the early detection of tree drought stress or forest stress in general. So as you may all be maybe well aware, our ecosystems face numerous challenges. They can be roughly grouped in 3 syndromes: climate change, that incorporates warming and also droughts, altered rainfall patterns and so on, land use change such as deforestation, urbanization and so on, and biotic threats, mainly pests and pathogens, but also invasive species. And my research focuses around that first group, climate change, and then the biotic threats. And in my research, I focus on trees and forests. And why? Simply because trees are magnificent creatures to start off with, but also because trees are huge players in the global biogeochemical cycles. For instance, forest ecosystems provide more than 50%, more than half of the terrestrial net primary production. So the difference between what is fixed in terms of CO2 via photosynthesis and what is released back in terms of respiration. So the remainder would be the terrestrial net primary production. But perhaps equally important, forests and the trees in the forest, they harbor roughly 90% of the terrestrial biomass in their vegetation bodies. That is a lot. So it's basically the best carbon storage, biological carbon storage we have at the moment. And that's why I'm so interested in forests and climate change or global change effects. And it all started during my PhD when we looked at the effects of elevated atmospheric CO2. And here I just provide a bit of a backdrop, a background for you. If you go back 800,000 years in time, and I think we have even further reaching time series in the meantime. And then you can see that the CO2 level, the atmospheric CO2 concentration, of all, like in the last million of years, has been in the range between 240 and say roughly 300 parts per million. And of course we have the ups and downs. Those are just the normal warm periods and ice ages as they occur on a semi-regular basis through time. If you ask yourself how we know about those concentrations. For those of you who are not too familiar with that area, those long time series are gleaned from ice cores, because the air bubbles that are trapped during ice formation, they are basically archives of the atmospheric air composition. And then, but back then in the 1960s, we started swapping those for exact recordings at Mauna Loa in Hawaii at this observatory. So we have this nice time series, and we can see here that after the onset of the Industrial Revolution, we have basically increased atmospheric CO2 by more than 50%. So we are at 421 parts per million right now, and it increases between 2 and 3 ppm every year. So that is quite a lot. And of course, that has an effect on our plants on this planet. Well, initially in the early 30s, when we became more and more aware of the increasing CO2 in the atmosphere, a lot of people hailed that or touted that as a greening of planet Earth, because CO2 is, of course, one of many prerequisites for photosynthesis. But it's not a one-to-one translation of higher photosynthesis into higher biomass. That is not true. And in the early to late 90s, a series of large-scale ecosystem experiments called free air carbon dioxide enrichment experiments were conducted, and my PhD took part in one of those in northern Switzerland, the Swiss canopy crane web phase site. So what we had there is we exposed parts of a natural forest, a mixed forest, that was about 100 years old at the time. We catapulted that forest into the future by fumigating it artificially with CO2, thus creating an atmosphere of high CO2 as we would expect it around mid to late century. And the way that worked, we had a huge CO2 tank. We used exactly the same CO2 that is used to produce Coca-Cola. It's fossil origin. And that CO2 has a nice property that has a very strong isotopic signature. So we can use it as a tracer. So it leaves basically a fingerprint in the ecosystem. And we can trace the carbon fluxes. And that CO2 was brought up into the trees via a net of laser-punched tubing. So very high precision tubing. And those released the pure CO2 into the tree canopies, and then we had sampling lines running down to a gas analyzer, so we could always check that we have the right or adjust the CO2 concentration in the canopy. So this is how we basically elevated the CO2 in the system, and my job as a PhD student was to measure leaf photosynthesis to see if after 7 years the initial stimulation in foliar photosynthesis was still there. So if elevated CO2 still brought up an increase in photosynthesis. And then I measured soil respiration to see how root and microbial respiration responded to the CO2 enrichment. And I also looked at fine roots. And then, of course, the overarching theme was also stable isotope analysis. And I just pick a few key findings of that study, which has been concluded in 2008. So after 7 years of CO2 enrichment, we had 5 tree species, but only Fagus, so beech, and Quercus, which is oak. They were properly replicated, and Carpinus. But if we look across all trees, and then we can see that photosynthesis, black means elevated CO2, white is ambient CO2, so the control. That during midsummer, the first 2 bars in July, photosynthesis is roughly 50% enhanced. And towards the end of the growing season, when the biochemical performance goes down a little bit, we still saw a 42% increase in photosynthesis. That was about 2 weeks before leaf shedding. So the leaves were actually going pretty strong. So there's definitely more carbon coming into the trees. And as I said before, that does not necessarily mean more growth. Because when we looked at the growth, these are tree ring series from the 2 best replicated species, beech and oak. The gray area here indicates the CO2 enrichment area, and this is the average over this area, and you can see white is the control, black is CO2 enriched trees. There's no statistically significant difference in radial growth of those trees. So quite puzzling, but not too surprising in the end. Even when we standardized the ring width by the previous 10 years to get a bit more of a stable signal, we could see that there may be 1 year, but it was probably more coincidence than anything else, where we found an effect, but overall nothing. No consistent growth stimulation under elevated CO2 in two of the most important, of the dominant tree species in Europe. Now one could ask, well, maybe they produced more litter, but the LAI, the leaf area index of that forest, the leaf area was already, as you would expect it from a mature forest, so there's actually not much space to expand more foliage. And that's what we saw here. We collected leaf litter with traps, and it was always the same story, no difference between the white and the black bars, so between control and CO2 enriched trees. So it didn't go, the CO2 that was additionally assimilated through photosynthesis under high CO2. It didn't go into growth, into stem growth. It didn't go into more leaves. I looked at the roots, but I spare you that graph now because it's the same story there. The trees didn't grow more fine roots. Then my respiration data showed actually some interesting results. Because what we could see here, the isotope signal, clearly shows the CO2 that we injected into the forest had a lower signature, and we can see that. So the CO2 made it into the soil. That's a quality seal up here. And then here we see the soil air CO2 concentration in the soil pore space. That was also elevated. But for some reason, it didn't want to come out. This is the soil respiration. So the soil that is released from the soil surface that we've measured. And that's a combined signal of what the tree roots are respiring and what the soil microbiome is respiring. And as you can see there, there's no significant difference yet again. We were asking ourselves, where the hell is that CO2 going that is fixed in photosynthesis? Well, one explanation at least for why I didn't see much more an elevated soil respiration was that a colleague of mine measured tree transpiration. We call that sap flow. And he found that trees that were exposed to elevated CO2 reduced transpiration. And therefore, more water was left in the soil. So we had soil moisture savings. But he didn't measure soil moisture. But when I measured, I knew his findings. I installed soil moisture sensors, and that is exactly what we've seen. You see the black line is elevated CO2, so the soil moisture around the trees that were fumigated. And the gray line is around the trees that were not fumigated. And the soil moisture is lower in trees that did not receive CO2. So there are definitely soil water savings. And you can also see that during winter time, it goes back to the same soil moisture here, and it gradually converges here. It's just during the growing season, when the trees are transpiring, that we see that difference. So that was the only really clear-cut signal that we saw, that elevated CO2 led to soil water savings. Overall, the take-home message was that we found no evidence of carbon limitation in terms of growth in those 5 tree species. What we did see was an increase in soil microbial biomass. So the trees funneled more of the sugars below ground. I mean, if we eat more, we simply get fat if we don't work out. But trees have the capacity to shed some of the extra carbon via their root system. So they can release carbon in the form of low molecular sugars, sugar alcohols, and so on, and feed it into the soil, and therefore feed the soil microbial community. And that, of course, stimulates soil microbial activity. It's a so-called priming effect, and it kind of increases nutrient cycling. So we suspect that long-term changes will happen in the soil carbon pools, but the soil carbon pools are inherently slow responding. So in order to detect such a signal, you would need much longer experience than those that run for 10, 15 years. You would need many decades to pick up that signal. We also picked up a little bit of an increase in dissolved inorganic carbon, so basically carbonic acid, if you want to, like, carbonated water. So a small portion of that extra CO2 dissolves in the soil water and is then either dissipated as runoff or leaches into the groundwater. So there's a bit of a mineral weathering increase perhaps involved. So that was that big first portion of my research career. From there, I went on to Australia, to Western Australia, a biodiversity hotspot, where I worked in banksia and eucalypt woodlands. It was quite a devastating experience because when I got off the plane, I was actually looking forward to being in Australia, and I knew it's a Mediterranean type ecosystem. The plants are used to summer drought, but I got off the plane and all I saw was plants dying, plants that are highly adapted to drought. So my a little bit unfortunate job was to look at why they were dying. So I did some work in banksia woodlands, and then I also did some conservation work with colleagues in a tuart, that is a species of eucalypt tuart woodland. That species is endemic. It only occurs in, I think, 25 square kilometer patch, so not very much, and it didn't regenerate anymore at all, probably due to climate change. But what we found through a nifty experiment was that soil ripping, so the soil is only sand, but it bakes really hard. It solidifies during summer, that if you crush that crust, and if you fertilize, then you get higher seedling growth and survival, which was quite astonishing and a nice restoration ecology outcome. Anyway, from there, I went to the New Zealand Forest Research Institute and got involved with tree pathogen research and also plant disease management, as you can see from the publications here. Most of the research revolved around a nasty genus of fungus-like microorganisms. The group, in English, the common name, I think, is water moles. The genus is called Phytophthora. And there are, I guess, in the meantime, close to 200 described species. Most of them are benign, but some of them are real, excuse my French, are real badasses. So they cause tremendous damage to either foliage or roots, and very often lead to the demise of not only single plants, but whole ecosystems or plantations. So we worked on different Phytophthora species, but mainly Phytophthora pluvialis, that caused great damage in the main plantation species, radiata pine in New Zealand. Did a lot of work there. Also did some work in native ecosystems. There's an iconic species, also a conifer, the kauri, Agathis australis, a Gondwana relic. Only occurs in the northern tip of New Zealand, and that was also ravaged by actually a native Phytophthora species that was benign up until climate change or global change kicked in, and then it started to kill off via the roots, to kill off that tree species more and more. Anyway, we had, with Peter Scott and Nari Williams, we had two Phytophthora specialists working at that institute. And this guy here, Peter Scott, was a hoarder, so he compiled more and more data of Phytophthora, different databases, created his own database. And at some stage, we were saying, look, let's just make that a global paper. We have so much data, I think we can exploit that database. So we we wrote that paper together, a highly cited paper in the meantime, where we looked at the global distribution, and we raked in some World Bank indicators, like economic indicators, population indicators, to model. So we knew what was there, and then we modeled the expected number of Phytophthora species per country. And then when you subtract your predicted or reported species minus the predicted species, so the residuals basically, they give you a really nice false color map of species that of countries that are potentially underreporting Phytophthora species. Well, let me just guide you through that real quick. So you can see that there are a lot of species reported mainly from countries that have a very intense agriculture and forestry industry, because this is where you have the problems with Phytophthora mainly. So North America, That's Australia, New Zealand, of course, Europe, China, India, and also a little bit in Brazil. And here we got our predictions, and then if we subtract those two, and then we can see everything that is going into the blue, those are countries that predict actually more species than you would expect. So they are doing a really good job. They're doing their homework. And then the countries that are getting more into the red, I'm not calling any names here, but they are expected to have many more species, but curiously, they are not reported from there. Of course, that would impair the trade business, so I leave the conclusions to the audience. But what we also did is we used the data from that database that Peter compiled, and I did some modeling. And at that time of that study, we had about 180 Phytophthora species formally described. And our estimates, the highest at the upper boundary of the confidence interval, we nearly doubled that number. So the expected number of Phytophthora species is somewhere between, rather towards 400 than only 180. And experts have confirmed that. So I also did some work on insects, mainly revolving around bark beetles and longhorn beetles that were attacking, again, radiata pine. But we also did one more general study where we compared LED lighting to conventional municipal lighting, high-pressure sodium lamps, the normal street lamps, because there was a huge transition from the conventional lighting towards LED back then, and we were asking, well, what are the impacts? There are impacts on wildlife, but no one has looked at that. Before we transition in a haste, let's have a study, let's have a look at what happens there. Now, to give you a bit of a background, a lot of insect groups have a high sensitivity in the blue region here. That means they are attracted heavily, strongly by blue light. If you look at the conventional street lights, they have a huge peak in the yellow. Yes, they attract insects, but just a normal amount. Okay, now if you have a lighting system with a huge output peak in the blue, then of course it's not surprising that you would attract more insects. And so this is what we got from the manufacturer, and this is what we got in our traps. So we caught nearly 50% more insects in traps arranged around LED lamp lighting than around high-pressure sodium lamps, the normal conventional lighting. That was a very nice finding, a little bit frightening as well, but I mean LEDs can be tuned, so you have to take out the blue light, very informative for the future lighting industry, of course. But what we didn't know was that in exactly the same year, just by chance, the Nobel Prize was awarded to 3 Japanese scientists, physicists, who invented the LED. And that's the same year as our study came out, so we kind of spoiled that success just a little bit. But yeah, it was a nice coincidence. Right about that time, I also got involved with some colleagues from the University of Arizona in a big meta-analysis that I performed, actually 2 meta-analyses. Those guys had a huge collection of soil samples from the Arctic, of different ecosystems. And we looked at temperature effects and also the shift from aerobic to anaerobic conditions, or the other way around, from anaerobic when the soils are waterlogged to aerobic conditions which can be expected under warming scenarios. And what we found is that, so common test in the lab is to increase the temperature by 10 degrees. And what we found there is that across the board, the ratio of soil CO2 release, soil respiration basically, increases by a factor of 2. So it's highly responsive to temperature. That means a lot of CO2 is released from those soils in the Arctic with warming, even if it's not 10 degrees. And also, you will, with an increase in temperature, a lot of the waterlogged soil will also become or transition from anaerobic conditions to aerobic. And that also causes an increase between, on average, 2.3 to 3.4 compared to the anaerobic conditions. So there's a lot of carbon potentially being released under global warming, which is really bad news because it will go straight in the atmospheric pool. Anyway, when I joined Auckland University, I kept my old ties to the research institute, Forest Research Institute. We kept on working on those topics, but I also did some more general plant ecological or tree ecological things at the tree line. And because I like to collaborate and I like data, I also worked with colleagues from the marine group in our department. And they had a very interesting study where they wanted to investigate the altitude of UAVs, or the impact of the altitude of UAVs on the behavior of bottlenose dolphins. Because UAVs were just coming up, and a lot of people were flying them, and they were trying to get as close as possible to the dolphins all the time. And then we did a proper study that showed very distinct behavioral changes if you flew at a height of around 10 meters. And a lot of private people flew lower than that. So you had to fly at least 25 meters or higher up so that the animals wouldn't be unduly disturbed. That was quite interesting. And then just before I left New Zealand, I became part of an international initiative. We just got our first paper out last year. It's called the MESI initiative. It is collecting, it's a huge database of manipulation experiments that revolve around terrestrial global change experiments. So it was a huge endeavor. We compiled multiple databases, and this is now ready to be used in all sorts of analyses, and a very exciting cooperation. And the second paper is in the pipeline. Then when I came here, I expanded my research a little bit, or maybe I should say I was roped into forest and tree nutrition by Johan Beyer and Carl Svensson. So I still do a lot of global change related stuff, but yeah, also the nutritional aspect is in there now. And even in New Zealand, I got really interested in hyperspectral imaging. And that's something I share with Johan Fransson and with Johanna Witzel. So that is also one of the research avenues we want to pursue here, using hyperspectral images or signatures from forest canopies to infer about the physiological and the biochemical status, possibly to use as early warning signals for pathogen or bark beetle attack or other stresses. And then I also work with a little bit with forest management effects on soil trace gas emissions with Charlotta, for instance. And yeah, I want to talk a little bit about, because we just got two hard-hitting papers out. I want to talk about one that just is hot off the press. It just came out last month, where we looked at early and repeated nutrient additions. And I know forest fertilization has not the best reputation. But what we do, at least scientifically, is not simply dumping fertilizer into the forest, but we prefer to call it nutrient optimization because it's a scientifically backed concept that is based on matching the nutrients that we bring out into the forest on what the leaves tell us. So we take foliar samples, needle samples, and look at key elements or the ratios of those elements to inform the nutrient, the fertilizer composition that is brought out into the forest. To give you a bit of a background, in this area of our world, the forests very often grow on inherently nutrient-poor soils, so especially when it comes to nitrogen. And therefore, forest fertilization is actually quite common, and traditionally it is applied very late in the rotation, usually a one-off application of around 150 kgs of nitrogen per hectare. And if everything goes all right, but more often than not it does not, then you would get a 30% yield increase. And that is across the board, across Norway spruce and Scots pine stands. However, the question is, or we know from the life history usually shows us, and the biological reasoning behind it, that the nutrient demand is usually higher in the early growth phases, so when the trees are still young. But no one had really looked at that, and then we compiled 9 different studies to look specifically into that effect, into early and repeated nutrient additions, and to see if that actually yields more stem volume at the end than the traditional method. So what we've done, we had 5 studies looking at a plot level, 2,500 square meters, at 3 different regimes. Nutrient additions every year, every second year, or every third year. And that is in green. Here is the control, so no nutrient additions here. Every year is red, every second year yellow, and then the dark green, bluish, is every third year. And what you can see here is that the growth, that's the annual increment in wood volume per year, that they're all growing much, much, much better under either of the regimes. And you can see that there's not much difference between the yearly and the 2-yearly fertilization. And at the end of the game, when you harvest, or at the end of the study, we can see that we had more than a doubling of the stem yield when we fertilize yearly or 2-yearly. More than a doubling. That is a lot of wood. And still 80% when you fertilize every 3 years. So there's a lot of potential to gain. Then the question was, well, what we see on the plot level, does that hold on the stand level? And then we used what we deemed practically feasible, a 2-yearly fertilization interval, and applied that on the stand level, 5 hectares. And there we saw very similar effects, also almost the same, very, very similar numbers, 114% increase, so more than a doubling of the stem volume yield at the end. And that shows us that the nutrient limitation severely restricts the potential to capture carbon and to form biomass in those spruce forests in this region of the world. Less than half the potential can be realized, and that repeated and early application of fertilizers can actually be an effective management tool, especially in support of a growing bioeconomy. And that got me very interested from a physiological point of view, because one of the hot questions at the moment is about the interplay between drought and nutrient supply. So I've put together a Cocoa project, and we got the industry that pledged support from Södra and IKEA and Sveaskog. So we hope that we can hand that in this year. And that looks at the role of soil nutrients in mitigating drought stress in trees. A lot of those individual forest nutrient optimization trials, they are spread across Sweden. And when I looked at the locations, I realized that they neatly, or some of them at least, you can pick and choose, they align along a latitudinal and longitudinal gradient. So we basically have a temperature and growing season gradient from south to north, and we have a precipitation gradient from west to east. And because the experiments are running or have been running for years or decades already, all we need to do is go there, take stem cores. We have a fabulous instrument here, an X-ray fluorescence densitometer. And with that, we can look at not only the growth, but also the density of the tree rings and the nutrient content in the tree rings. And with that, we can hone in on drought effects. Like, for instance, you all remember the 2018 horrendous drought year. We can go back in time and look at those effects and compare the trees that have received extra nutrients versus those that have not. And then against the backdrop of the temperature and the precipitation gradient. So a fantastic playground, basically. And that can be complemented, of course, with drone hyperspectral imaging. Because one would assume that even under the normal summer drought, mild summer drought that the trees see every year, we should see a difference between the trees that have a higher nutrient supply as opposed to the ones that don't. Anyway, and that opens up a whole, can of new research avenues. We just got granted an EU Horizon project, Opti for Value, worth 5 million. About 10% is flowing towards LNU. And we're going to look at some of those aspects, mainly revolving around the early detection of biotic and abiotic stress. Right. Do we have time? We have time for one more, perhaps, pet project of mine. And that is something I want to tackle this year, and it revolves around the transpiration control in trees. So the transpiration, the gaseous water loss from trees, is mainly driven by a meteorological parameter called vapor pressure deficit. It's basically just atmospheric dryness, a measure of atmospheric dryness. And global models have shown that this vapor pressure deficit is increasing, as does global warming. So it's driven by global warming. So we have an increase in this vapor pressure deficit. As a bit of a background, we need a certain amount of, let's just call it dryness in the air, a certain amount of vapor pressure deficit to drive transpiration. If it is very humid, the trees do not transpire a lot because the gradient is very low. So you need dry air outside. The air in the leaf is very moist to have a bit of a pressure gradient, to suck the water basically out of the foliage. So you need some level of VPD to get transpiration going. And when trees transpire, they lose water in the form of water vapor through little pores on the underside of the leaves or the needles. They're called stomata. And those pores provide a two-way avenue, one for water vapor out, and the other avenue is CO2 in for photosynthesis. Okay, so that's the background. If you have very dry air, plants lose a lot of water, but they have the possibility to regulate those pores so they can open and close. Stomatal regulation. So I told you we need a certain amount of VPD or a certain amount of atmospheric dryness to get that process going. You can see that here. We have a very high stomatal conductance. There's a lot of water passing through those pores, but as the vapor pressure deficit increases, the transpiration goes down. And with transpiration, as we've just seen in the previous slide, also the carbon fixation and hence the ecosystem productivity. Now, this is across ecosystems, but it's not the same for each species. And we did a study in Switzerland where we compared trees with different wood anatomy. So we have 4 broadleaf species here, linden, beech, maple, and cherry. And then we have oak and ash. And those 2 are a bit special because they have what we call ring porous wood. They form very wide vessels and conduct a lot of water early in the growing season, and they have usually very far, very deep-reaching root systems. So they're connected to deep-lying water reserves. And that's not the case here. Diffuse pores, they form similarly sized vessels across the tree ring, and the root system is not as deep as in those 2 species. Now what we see here is the transpiration. We call it sap flow as a color scheme. So very low is red, very high is blue. And on the y-axis, we see the soil water potential. It's the soil moisture, basically. And that is good if it is low, and if it's strongly negative, if it's strongly negative, then it's very dry. If it's going towards 0, that's very moist. And down here we have the vapor pressure deficit. Again, 0 is not good because then it's not driving any transpiration. But if it's too high, then the plants start down-regulating the stomata. And then the lower row is in a moist forest, and it's in a dry habitat. So we can see the most sensitive is linden. It quickly down-regulates stomatal conductance and transpiration. And they all do pretty well if there is a lot of moisture, so high up here, and a decent amount of VPD, so between, say, 1 or 1.5 and 3. But what struck me was that the 2 ring porous ones, they seem to very, very quickly down-regulate, no matter how much water there is, no matter if the forest is dry or moist. They seem to always quickly down-regulate their transpiration to about 60–70% of the maximum. So they seem to be super conservative, which makes sense because they have those large vessels and they cavitate quickly, although they have connection to deep-lying moisture reserves. But that struck me. And when you compare the 2 habitats, you can get a ranking, and that shows that the 2 ring porous species are the least sensitive to soil drying or atmospheric drying because they have access to the deep-lying soil water resources and they sharply regulate transpiration. Very conservative. And then for me the question was, okay, so apparently they start regulating transpiration very early on. So what we usually do in physiology, we have those response curves. And then, of course, everyone sees that the transpiration is leveling off with increasing VPD. But when exactly that happens? Initially we have a linear increase, which means the vapor pressure deficit, the atmosphere, is the only driver for transpiration. But when it starts leveling off, it means something else is controlling the transpiration. That is the stomatal conductance, the leaf pores. But where exactly that happens is difficult to gauge and is prone to large errors. So I came up with just taking the first derivative of that curve up here, and that gives you the exact point where the curve deviates from linearity. So when stomatal regulation kicks in and takes over more and more, and the curve starts to deviate from linearity. That is the workflow. Not super important how I do that. The most important bit is that when we plot that, we indeed see, at least in ash, that the downregulation of stomatal conductance indicated by those lines occurs earlier at a lower vapor pressure deficit, as expected, compared to the diffuse porous trees. So there are marked differences, in that case linked to the wood anatomy. Now the big goal or the big challenge is to exploit a global database of sap flow. So this is across all biomes and all different wood anatomies, and also angiosperms against conifers, to basically milk the database, exploit the database, and apply that new metric that I've come up with to the database to see to get a worldwide comparison in those in this critical VPD, where the environmental control of transpiration shifts towards biological control. Yeah, and I skip over that. That would be it. Thank you for your attention.